dear students of second year D Pharmacy, third year B Pharmacy, and the final year B Pharmacy. Welcome you all. I welcome all of you for this program Rendezvous 2021 for this Industry Expert Lecture 2021. Welcome you once again.
Sathe sir.
Sathe sir is not here, sir. He's just gone out, sir.
I think Virgesh sir has disconnected.
Sir, बाहेर गेले, sir.
Sathe sir.
Hello.
Yes, sir.
Sir, interruption was there, sir?
Yes, sir.
Okay, sorry for interruption. Let me start with begin. Okay. Yes, it is visible, sir, all of you here?
Yes, sir. Yes, sir. I can see you.
Yeah. Okay, thank you, sir. Okay.
Sir, guard sir आहेत.
Okay, just a minute, sir. Sir, but limit is over. I think 100 is there, sir.
आहे sir. Shrikant sir आहेत meeting मध्ये.
Shrikant sir आहेत. Yes, sir. Just I'm
okay.
admitting. Yeah. Okay. आहेत sir. Already आहेत. Yes.
Already आहेत.
Yeah. Okay. Thank you. Okay. So sorry for interruption. A technical issue was there. So good morning to one and all. I welcome all the dignitaries present over here for this program Rendezvous Industry Expert Lecture 2021. I welcome Honorable Chief Guest, Honorable Mr. Achyut Sawant Bhosle Sir, Executive Chairman, Sri Yashwantrao Bhosle Educational Society for this program. Welcome you, sir. I again welcome invited speakers, guest, industry experts, Honorable Mr. Veer Dhamal Kamde Sir, Deputy Manager, Quality Assurance Department.
सर गौतम सरांचा परत डिस्कनेक्ट झालाय बहुतेक. सर
मी गौतम सरांना माझ्या एकटीच बोलून घेतो.
चालेल सर.
Hello, Gautam sir.
Hello.
Yes, Gautam sir.
Yes, sir.
Please proceed.
Yeah. Again, I heartily welcome Honorable Mr. Shrikant Gad sir, QA Executive, Glenmark Pharmaceuticals, Goa. Welcome you, sir. I again welcome Honorable Dr. V.A. Jagtap sir, Principal, Yashwantrao Bhosle College of Pharmacy, Sawantwadi. Welcome you, sir. Again, I would like to welcome Mr. Satyajit Sathe sir, Principal, Yashwantrao Bhosle College of D Pharmacy. Welcome you, sir. Again, I welcome Mr. Omkar Pense sir, HOD, Integrated Diploma Pharmacy, Mr. Tushar Rukari sir, Head of Department of Pharmaceutics. And I welcome all the participants, dear students of second year D Pharmacy, third year B Pharmacy, as well as the final year B Pharmacy. So welcome you all. So objective of this today's program, Rendezvous 2021, to promote the interaction between the pharma industry and academics, to bridge the gap between the curriculum of pharmacy and industry, and to interact the industry expert and the students to explore the pharmacy students with the current scenario of pharmaceutical industry. So dear all, As our tradition, any auspicious work, we should start with the lamp lighting ceremony. So I request all the dignitaries uh, present over here, please proceed for the lamp lighting ceremony. So,
सर सॉरी थोडासा
sir, voice is not there. Yes,
सर थोडा प्रॉब्लेम झाला चेंज झाला स्लाइड
please, huh?
आय विल स्टार्ट सॉरी
Yeah, please, huh? Sir, voice is not. Yeah,
Please
thank you.
proceed forward.
Yeah, yes, sir. Thank you all. Now moving ahead, I would like to request Honorable Dr. Vijay Jagtap sir, Principal Yashwantrao Bhosle College of Pharmacy, Savantwadi, please share your valuable views about this Rendezvous 2021. Over to you, sir, please.
Thank you, Durgesh sir. Very good morning to all. Uh, Yashwantrao Bhosle College of Pharmacy and Yashwantrao Bhosle College of D Pharmacy, uh, jointly organized venture. Rendezvous is that concept where we are interacting and we are inviting industry experts who interacting with a student and guiding the things what's actually the industry needs. uh, as we know that everyone is talking about the quality, so quality it's one of the major point nowadays, important in every pharmaceutical industry. So if suppose the current scenario, if you go through that, industry expect such type of students who will be dedicatedly works towards the quality of the medicines. And it's very fortunate that we have started this series, Rendezvous series, since from the 2018. And we have been got the cooperation from various industry experts from the production, quality control, quality assurance. And today it's also, we are very much pleased to welcome all the dignitaries from the Glenmark as well as Centaur, particularly Guard sir, Nanoskar sir, and Kamble sir. So this will be a feast for a student. Definitely, they will get more idea that what type of work is going in the industry, and what type of employability should be developed by these students uh, within these coming years. So they can be absorbed very well nearby in the uh, Goa or some Mumbai region, where pharmaceutical industries are in boom. But one of the major problem is only one that industry experts expects that the students must be lab oriented, they must be research oriented, also they must have some basic qualities. And definitely I'd like to wish all the
Yeah.
guests, I would like to request all the guests that
Hmm?
they should guide a student related to this point of view. And after all, I would like to thank training and placement cell of both the colleges, particularly Gurish Gautam sir, as well as Mr. Omkar Pense sir, for organizing this event amidst COVID. And we are organizing, this is the first time this event online, virtual, and students are also taking the efforts. So I wish training and placement cell as well as Mr. Satyajit Sate sir, principal of Yeshwantrao Hostel College of D Pharmacy, for organizing this event. And I thanks to the management also, who is cooperating every year to organize such type of events. And I wish at the end very grand success for this whole day event. Thank you very much, sir.
Yes, sir. Thank you so much, sir, for your valuable words to for your our students. Hello. Hello. Yeah.
Yes,
Yes.
Yes,
yes,
sir.
sir.
I would like to request Mr. Satyajit Sate, sir, Principal Yeshwantrao Hostel College of D Pharmacy, to say few words regarding this program. Over to you, sir.
Okay, thank you, sir.
Yes, sir.
Hello, good morning, all. Wish you all the best for this today's session. As Gautam sir said, now this is a need to bridge up or to fill up the curriculum gap between the between syllabus and the today's need of industry. So arranging this type of lectures is very important to fill up the gap between curriculum and industry need. So because of this, we are going to start this event, or we are going to organize this event under the guidance of our executive chairman honorable Achut Bhosle sir, as well as our beloved principal honorable Dr. Vijay Jagtap sir. Due to his constant support and due to his constant guidance towards us, so it is possible to organize this event. On the behalf of Yeshwantrao Hostel College of D Pharmacy, I am welcome all to today's guest speaker, Mr. Nanoskar sir, Mr. Shrikant Gad sir, and Mr. Virdaval sir. Heartily welcome to this program, and I hope this program will be very successful. Okay, thank you, sir.
Yes, sir. Thank you so much, sir, for your valuable words. So now, before proceeding this first industry expert lecture, I would like to share some brief information about our today's guest, eminent person, industry expert, Mr. Sandeep Nanoskar, sir, Quality Assurance Executive, Center Pharmacy, Goa. So, Mr. Sandeep Nanoskar, sir, he has done Bachelor in Science, BSc, from the University of Mumbai in 1993. He has done Masters, MSc in Organic Chemistry in 1995. Mr. Sandeep Nanoskar, sir, presently working as a Quality Assurance Executive at Center Pharmaceuticals Private Limited, MAPSA, Goa, since from 27 July 2009. Previously, Mr. Nanoskar, sir, he has done work as a for 5 years as a quality control officer in Cosmo Pharma Limited, Bicolim, Goa. Also, Mr. Nanoskar, sir, he had worked for 2 years as a quality officer in Maya Rasayan Limited, Chipdun, Maharashtra. So, current job profile of Mr. Nanoskar, sir, he is reviewing of finished product reports, bulk reports, raw material reports, packaging material reports, analytical method, validation, calibration of some instruments, all documents related to the data integrity matter like a result of out of specification, out of train, system stability, failure, laboratory error, and incidents. So all these are the some current profiles of our today's guest, Mr. Nanoskar, sir. Then he has also handling the some method development, reviewing the training and records of the chemist. So now I would like to request our eminent speaker, Mr. Sandeep Nanoskar, sir. Please proceed for today's your lecture. Over to you, sir.
Can I proceed, sir?
Yes, sir. Yes, please. Over to you, sir.
Welcome to all of you, sir. Namaskar to, because I am a Hindu, I will say namaskar to all of you who are joined here, the students, Princess sir, Guard sir, Satri Sate sir, Jagta sir. Today's my topic is good manufacturing practice. Can I share some slides, sir? Gautam sir, I want
Yes,
to
sir.
share
Yes,
some
sir.
slides
Just
with
I'm
the
yeah.
people
Yes,
who
sir.
are
I'm
joined
giving
here.
yeah. Yes, sir. Just I'm giving
How
a
can I share, sir? Sate sir, please help me.
हॅलो गौतम सर.
Yes, sir.
कारण
I have.
की आहे
Yes.
सरांना होस्ट करा, सर रन करून घेतील.
Yeah, please.
Yes, sir.
You
मी
can
stop
proceed.
करतो sharing, sir.
Yes, sir.
गौतम सर.
Yes, sir.
सरांना
Share
होस्ट
this.
केलं तर सर will able to share the PPT.
No, I have just here. Multiple participants can share the simultaneously. I have mentioned, sir. I have click over there. So, sir
Now,
can.
now I'm trying to share.
Yes, yes, sir.
Yes, sir. Please. Yes, sir.
Yes, sir. Can you see the screen of
Yeah.
launcher, sir?
Yes, sir. Yes, sir.
Yes,
It is
sir.
visible.
Okay,
Yeah.
no
Thank
problem.
you.
You
Yeah.
can
Yes,
start
sir.
the session.
Okay. We'll start our topic, please. My topic is good manufacturing practice because Lad sir and I think Naik sir will go for quality control implementation and current senior of quality control. So I will prefer to talk one good manufacturing practice. That word itself clears that the manufacturing practice should be in good manner. Manufacturing practice should be in good manner. What is the GMP? Good manufacturing practice is a set of regulations, codes, and guidelines for the manufacturing of drug substances, drug products, medical devices, and foods. What does it mean? Today we are seeing, we are experiencing from last 1 year, 1.5 years, so Hindi culture was followed by all the people in the around the world. Okay, the US president also following our culture, so our regulations, our rituals, our what we call the dharmic vidhi in Marathi, no, rituals are followed by most of the people in the world. So we are talking here. We will talk about the GMP in the pharmaceutical field. There are some regulations, there are some codes, there are some guidelines within the company. Various company has different guidelines. If we go for the regulation, we go for audit. We have to follow the guidelines, the codes, and the regulations also. What are the regulations? The Lard sir or Kamesh will explain better than me because he has worked in good company like Glenmark or other companies. I'll give you the brief idea in this. Regulations means there are some regulations, no? There are rules and regulations which we are following day by day, day to day life. You know, our college, you are college also, you are following the regulation, no? You are entering the on gate, you are coming, you after completion of your lectures, you are going outside the company, outside your premises. That are the regulations. What are the codes? The government has quoted some codes for different processes, for quality control, for quality assurance, production, for warehouses, for safety and hazard. These codes should be followed by each and everyone in who are working in the premises of company. Okay, the guidelines also there. The different guidelines are there. In QC there are different guidelines. In production there are different guidelines. In warehouse there are different guidelines. In quality assurance there are different guidelines. And there is even some sequence. The sequence should be followed by each and everyone who are working inside this premises. The main objective of GMP is to guarantee high quality product to the customer. Today we are facing new problem now, corona or some diseases like corona or other diseases who are out, which are not curable. So guarantee high quality products. Our every action of every personnel who are in the company, who are working in the company, should be like that. The customer should get the product which is free from all the possible contamination. There should not be any type of contamination. What does mean contamination? There are different things. The personnel who are working should be have trim nails. He should be take personal hygiene like that. Okay. A general requirement: avoid risk, possibilities, or mix-up at all stages of manufacturing, labeling, packaging, and testing. Avoid risk. Everybody should avoid risk. If somebody say a sampling person is there, new sampler, new chemistry join in company, and expert person is also there. The new person comes directly go for sampling. Here to if he is sampling, no, he have to change the gloves, he have to change the spatula, he have to change the sampling tools at every step for every material. But the new people, what they do, they are not following the all the requirements which should be there. Hmm? Labeling also there. If suppose sodium hydrogen phosphate is material is there and disodium hydrogen phosphate, so both the material came in the company in the store simultaneously. New person is not able to differentiate the two raw material. He may have chances, he may make mistake. He may label the raw material wrongly, which will harmful for the production and also the customer. Packaging also there. Packaging should be properly. There is a we say the grammage. We can say the pouches are there. They should be in good quality. And the testing related to quality control. All the taste of that raw material. Suppose if one raw material is came, with same batches came twice in a day, the person who is new in the quality control, no, he can omit the test in the raw material, and he will release the material which should be not it should not be there. Okay. AHU, air handling unit. I mean, I want to say the air conditions, comfort of personal working. The person who is working in the company, he should feel comfort throughout the day. Regular monitoring of temperature, humidity. Suppose in quality control, temperature requirement is different, humidity requirement is different. In microbiology section, there is requirement is different. The monitoring of temperature, humidity should be there after every 4 hours. The 3 hours, as per the protocol of company, the monitoring of humidity, monitoring of temperature and humidity should be done. The particle count. Another word is the particle count is related to the microbiology section. The particle count should be less. In the microbiology section, there should be list particle count that is there. The proper drainage system which prevent backflow. The drainage system should be in such a way that there should not be any type of backflow. If backflow is there, it should be have the provision of opening and cleaning and disinfectant. If the backflow is there, obviously the harm will be for person who are working in the company. Okay. The building and facility, how the building and facility should be in the company, in the pharma company, that is the main role also. The design and construction features, lighting, design of the construction, design or the building should be constructed in such a way that there should be demarcation in every section. Suppose in store, there should be demarcation of raw material, the finished product, in process or other material, we are rejected sample. There should be demarcation. There should be provision of separate rooms in the premises. Okay, lighting should be proper. Anybody in the premises can see the label, can read the properly. That should be the lighting procedure. Okay, ventilation should be proper. In some company, they are not providing the proper ventilation, which is hazardous for the people who are working inside the company. Suppose in quality control, many types of acid are used in the quality control. If the fumigate is not working properly, then it is very hazardous for the health of the people who are working in the quality control. So there should be proper ventilation should be there. Air filtration, air heating, air cooling, it should be proper. Plumbing, in our house, we are doing plumbing. No, same as the plumbing is done in the pharmaceutical company. The plumbing should be very prompt. As per the requirement in quality control, there is different requirement. In production, there is requirement different. In our washroom, requirement different. So the plumbing should be in proper way. The sewage system, should be proper. As I said earlier, washing and toilet facility. Washing and toilet facility, there should be washing and toilet facility for the every men and women should be in proper way. It should be away from the production area, quality control area, or the store area. It should be away from these departments. Sanitation. Now we are very much aware of sanitation. Everybody goes for sanitation. So sanitation should be proper before entering in the company. Now most of the companies have the provision of we have to wash our feet. We have to change our footwear, road footwear, and we have to go inside and then we have to go for the thermal scanning, and then we enter for our department. Okay, the maintenance of the maintenance the company, the maintenance department or maintenance team should be in, should able to sorry, should be able to overcome any problem in the company, and it should be in very fast way. If suppose the light is gone, tube is gone, it should be changed very fast. In some company they are using autoclave. If the autoclave is not working properly, then it is not suitable for the health of the company. Okay. The good management practice, a basic principle of GMP is that quality cannot be tested into a batch of product, but must be built into the each batch of product during all stages of manufacturing process. What does it mean? Suppose on company, if some test is on GC, after in some company they are validating their method. They are not doing every test on the GC. Suppose Olata company, OVI is there, residual solvent is there, or some tests are there. We are not doing every test of every material. After every first batch, 10th batch, 15th batch, 20th batch, 25th batch, we are doing in one annum. In some company they are doing all the tests as per the vendor's requirement. Okay, so we can omit the test of raw material, but in the production, in the formulation, we cannot omit any type of test. We have to do every and each process, each test of the formulation, that means the product which is goes to the customer. Okay, the building is designed to minimize the risk involved in any pharmaceutical production that cannot be eliminated through testing the final product. There should not be any type of risk, either in manually, either in instrumentation, either the electricity, either in the what do we call the building. The building should not have any type of risk. If some joints are there which are not filled, there is a chances of ants goes inside. If there are cracks in the wall, it should not be like that. The cracks should be filled immediately after they have happened. Okay. Now we are going for the GMP category. What are the categories in good manufacturing practice? It may be repeated, but it is very much important for the who are working in the company. The pharma, just now I talked to Mr. Sate sir. He told me that about 640 students are passing out every year. No, so for these people, there is not sufficient jobs are available in the pharma company. Somebody has to do work in the pharmacy. I mean medical store. Somebody have to go for a lecture. But the overall how the company works, that we should know. Okay, the premises, equipment, personnel, raw material testing. These are manufacturing control, packing material testing, finished product testing, quality control department. All these are comes in the quality control. Sanitation, storage system, stability sample, warehouse, records, and sale. We go one by one. Sorry. How the premises equipment, the premises should be like that. It should have the proper way of cleaning. When the person in the company gets some problem regarding the health, he should go to the change room, he should go to the washroom, and he should have proper way of cleaning or other things. If something happen in the quality control, then he have to wash his eyes, he have to wash his face, and he have to go for the next treatment. No, so there should be proper way of cleaning. Prevention of contamination. There should not be any type of contamination. As I said earlier, the contamination, there are various types of contamination in the company. There should be no any type of contamination in the company. The company should have restrict who are making the contamination. The ordinary condition. There are some orders in the company. The material comes in the company, it should go to the security, then the store, then the store makes the MRC, that means the material receive challan. It goes to QC people sample the raw material or the excipient, whatever it is, and then it goes for testing, Then we release the sample in quality control. Then we approve it for the weighing. That means the formulation people. I mean the store people, warehouse people, and the quality assurance people. They are weighing for the batches, and then the production people takes a batch. Again, the samples come to the QC, and that is called bulk sample. Again, the bulk sample release. Again, it goes for the packing or the filling. There are various stages, and then again, the last sample is again tested by the QC for the whole testing. Okay, the good state of repair, that is I have covered in maintenance. Okay, the personnel should be appropriate education. Who are joined in the company, who are joined in the company, so he should have the proper way of education. If it is BSc, MSc, or B.Pharm, M.Pharm, D.Pharm, he should have proper education. How to work in the company, how to work in the quality control, how to work in the quality assurance, how to work in the production. If he is for the packing department, how the packing is going on, he should be very much aware of that packing. He should be trained by the senior people. The senior people should review all the activities of the new people. He should sign, and then it goes for the approval. And the experience is also there. He should have proper experience. Sufficient number of people. If suppose production people require is one batch is to be taken for 500 kg batch, the 10 people are required. There should be 10 number of people in that team. If we want to complete our production in 8 people, then it will be either it will be delay or either it will be harmful for the batch. It should not be there. All the concerned people who are working should have the proper GMP training. The good manufacturing training should be given to all that, and concerned document should be made to go for the audit or other purpose. Okay. Sanitation is, I think I have discussed sanitation program to prevent the contamination. Sanitation means we are doing the fumigation in microbiology section. We are doing fumigation in sometimes in the physio also. The fumigation should be in weekly or after 15 days of monthly. The program as per the protocol of the company. Limit of sources and types of the contamination. There should be minimum sources of contamination, either in manually, either or by mistake of the personnel. Either if we receive the sample in not in a proper manner, then we have to intimate our the senior intimate to HOD, and what remedy should be taken. That is there. The cleaning procedure for facilities and equipment. After suppose I have our product is Cosset G, we have manufacturing Cosset G cream. It is just one of our products. If we have to take a second batch of Cosset, suppose the batch number is BP001, where in the evening in the second shift we take BP002. Then what the people are doing? They are not cleaning the facility. They are not cleaning the storage tank. They are not cleaning the manufacturing tank. They are not cleaning the equipment instrument which are used for the previous batch. They directly take the batch in that vessel. It should not be there. All the vessels should be clean. The line should be taken. It should be tested by the quality control people. The quality control people approves for the next batch, and then the new batch is taken for the production. All these activities should be done in proper way. Any bypass should not be there. That is the way. Equipment, cleaning of equipment. Suppose there are various types of equipment. As I said, in production there are manufacturing tanks, storage tanks, other pipelines are there. In quality control, the HPLC, GC, UV, IR, that reometer is there, viscometer is there, pH meter is there, conductivity meter is there. All should be cleaned properly in proper way, and the training should be given to the concerned people. The pest control should be done after every 15 or 6 days as per the protocol in the company. The environment monitoring means what? Suppose the requirement of the premises, requirement of the department 25 degree and 30 degree RH relative humidity, it should be like that only. The 2% difference is allowed. It should be monitored by the expert and by the HOD. If it is not there, in the environment of the in the department is not proper, then we have to go for the remedial action. We have to do the proper environment condition which is required for the activities. These are the this slide is about the testing which is done in the quality control. Raw material testing is there, packaging material is there, finished product testing is there. As I said earlier, each lot and each batch of raw material should be testing. If suppose B001 is raw material is come, suppose NaOH is come, sodium hydroxide, one batch number. If the second batch in the evening, sometimes because of transport problem, because of transport delay, the same material of same batch of different vendor comes in the our store, then we have to sample that material also. If the sample is of same raw material, I think same batch number, we have to sample twice. We have to do all the testing required in the required as per our requirement. Okay. To confirm the identity of raw material, we have to confirm the identity of raw material, whether the hygroscopic is there or we have to go for the identification test. Suppose as I said, sodium dihydrogen phosphate and disodium hydrogen phosphate. We have to go IR or pH. The pH difference or IR are very much there is difference in IR and pH meter. Okay, so we have to confirm the material which is desired to come is that or not. Provide assurance that the quality of the drug in formulation. This is the main thing. Whatever drug we are using in the formulation, whether it is effective, whether it has the therapeutic use or not, that should be confirmed. If the quality of the material is not that much, which is supposed to be, then we have to do any remedial action against them. Sample of incoming material are collected and tested before use. This is I discussed the material which is comes in the company, comes in the product store. Now we have to collect that and test it before every use. Approved and validated method and specifications are used. Approved and validated method, the method which is for suppose the production people are taking the batch at 500 kg, the manufacturing tank is of 500 kg. Then we withdraw the sample from bottom, from top, and from the middle of from right side, left side. We collect the sample. If we collect the sample, the QA person carries the sample to the QC, and QC people does the testing of that bulk sample. If the bulk sample is suppose the pH limit, the certain limit is 5 to 7. If the pH of the top sample is 3 and the bottom sample is 5.5, the bottom sample is okay. What about the top sample? It is acidic. If it is 3, it is very much acidic. Then the product is not but homogenized. Then we have to mix, we have to intimate the production people to mix it properly, to homogenize it, and then again we go for sampling and again do the validation. The method should be validated like that. The analytical method also there. The analytical method should be validated. There are different parameters which I cannot, which I am do not want to discuss. There are linearity, accuracy, specificity, ruggedness, robustness. That is not my subject of discussion. So there are various types of AMVs, analytical method validation. Result must conform to specification for release or for use or sale. Whatever result we are giving, suppose the result of the assay of the some drug, suppose there is ocupol, we are manufacturing ocupol, the active ingredient is chloramphenicol, be pure USP or IP. The release limit is 95 to 120. Our result should be within that. Our result should be within that. If the assay is coming 90%, we cannot pass the batch, we cannot supply the batch, we cannot dispatch the batch for sale. If it is sold outside, and if the customer, that means we may also the customer, we do not get the effectiveness of the drug, it is very big offense. As per the Cosmetic Drug Act, it is not proper. The transportation and storage record. The transportation also plays important role in pharmaceutical field. If suppose the transportation is not good, some workers are throwing the container. If the container get damaged, if the plastic bag inside it get distorted, then we have to examine that batch for various tests. We have to intimate the store manager or store officer who is there, who is working there, and we have to make any remedial action against them. The storage record. The record should be stored properly. Suppose the batch expiry is up to 2 years. As per the company protocol, there are different ways of storing records. There are different documentation room are there. The records should be stored properly, and it should be available very easily. Suppose the auditor comes in the company, he wants to see the record BPR, BMR, I mean batch packaging record, BMR batch manufacturing record, or COA certificate of analysis, or the any other report of raw material, any other report of the author. There are different types of author. Then we have to present our report to the auditor very immediately. Okay. How the manufacturing control can be done in pharmaceutical field? There should be written procedure in the company, and the procedure which are written should be followed by each and everyone. Supervisor, the supervisor should look after the every activity in production. If some new people join, he may take, suppose the we are manufacturing phosphate GM product, the micronazole nitrate slurry, we are adding at the last of the last stage of the manufacturing, and then we go for the batch size, 500 kg, 400 kg. If the new people is join, he may add the slurry initially. It is not proper. It is a one example I am just giving. There are various types of example in this. So the procedure should be followed by each and every one of this company. The master formula, manufacturing order, packaging order should be in proper way. There are the master formula in the various companies. So we have to follow that formula. Manufacturing order is also important. The manufacturing order should be followed in that way. And packaging order, critical procedure are validated. If the procedure which are very much critical should be validated by the expert, and in this validation, the QA person also plays important role. The QA person, the production in charge, the QC head, or the QC officer, QC executive had different types of role during the validation. Second line is every second person verification of activity. What does it mean? The second person of verification. If the person every person has very much we have a natural habit of escaping some step. No, so he can escape one step. Then the verification should be done by the personal. If it is done by the new people, who have to we have to understand him, and we have to tell not to avoid this test. Verification is very much important. And last one is self-inspection program. That will be internal audit. The internal audit should be planned in such a way that there should not be any discrepancy during the various activities in the production, in the quality assurance, in the product, in the quality control, in the store, in the different. So internal audit is done by the people. For internal audit, if the QC audit is going on, the QC person is not included in the internal audit. The other person from production or the other departments are included in that team. Now I will talk about some quality control department activities in quality control department. The testing of bulk component prior to use of prior to use by production. When the production people takes a batch, they sample the bulk sample. QA people sample the bulk, and it goes to the QC. The QC person, the pH is there, AC is there, 2 or 3 tests is there. For tablet, there are various tests are there. For tablet, there are various stages of bulk sample are taken. So it should be tested in proper way, and we release the batch for next step. It is the role of QC. Testing of finished product prior to sale release for sale. Every test of finished product should be done by the company. If the company is not able to do that finished test, then the company send the material to the public laboratory, which is accredited by NABL or some regulatory body. They have the authority to do that test. We get the report in our valid period, and then we release for the finished product. But the report of that test should be issued prior to the release of for sale. We cannot send our finished good in the market without having COA. Okay. It's a stability program. We are very much aware of the expiry date. When we buy some medicine in the company, pharmaceutical, we are just go for the expiry, within the 2 years, January 21, January, whatever it is. We tell that owners, we want to change this. I have to change this tablet. I have to have expiry update date very much larger. Okay, the stability program is there. Suppose the 24 months is the shelf life of period. Suppose the today batch is taken on 18th of or 21st of February. So the 2 years validity, that 2 year expiry date means was January 23 will be our expiry date. And during that 24 hours, our sample is tested after every period. There is a protocol for every company. Sometimes it may be common, sometimes it may be different. For 3 months, whether our formulation is stable for our desired use or not. After 6 months, we are testing. After 9 months, we are testing. After 12 months, we are testing. After 18 months, 24 months, like that. If the expiry date is more, then if the protocol changes as per the expiry date. But testing is done in various stages. In some companies, in our company also, we are testing the stability sample for 25 degree and 60 degree, 30 degree, 75 degree RH, 40 degree. This accelerator time is there also. For 6 months, we are doing the study. We are studying the stability sample for 6 months. That is there. So stability of our product should be proper. Our product should be very much stable. It should not give less effectiveness during the time of expiry. That is there. Review batch records and labels. If the batch records are made, labels are made by the anybody. If I made a label, I am prepared the batch record. It should be reviewed by some other person. It should be approved by some other person. There are at least 2% are there which are reviewing and which are approving the every record. That should be there. It is not that we are not trusting the people, but our human being, as a human being, we can make a mistake, no? So we have to review every batch, every label for the safety of the customer. Okay. Release of raw material, product based on QC result. The QC plays important of the in the company. The QC person has to release the raw material after completion of every test, after assay, after taking water content, after checking the pH, or various tests are there. We have to complete the test. We have to submit the report to our supervisor or reviewer. Reviewer checks the report. If there is a discrepancy, he may he may go for as I say, heard by our Durgesh sir, OS is there, OT is there, system suitability failure is there. If it is not there, then we go for the release. Training and auditing, the training of people in the quality control, we should train the people in proper way. Every newcomer should be trained. We have to document the training. We have to train the people in proper way. Whether it is after training, competition training, if we made a mistake again, then we have to go for the raise the incident or some other thing, and we have to send that person to the training, and again the experienced person may have to go for training. Auditing is also there. Auditing means different. There are many types of auditing. IS auditor there, WHO auditor there, US FDA, the Australia TGA is there, MHRA is there. There are various types of auditing are conducted. Are conducted in the company. The customer complaint. If suppose our company manufacturing the 5 gram of cream. If the customer is not getting 5 gram of cream, he is getting only 4.5 gram. It is a big offense, no? So where if the customer raises the complaint, then the complaint goes to our company. We, the manufacturer of that drug, he have to file that customer complaint and go for the other activity. In that case, no, the company may have take action against the people who are related to that product. So we must be aware of every action. The records, documentation of all the GMP activities should be done. That is also important. Whatever we are doing, we were entering here, no, we have to make entry. We are weighing, we have to weigh if suppose raw material weighed on the precision balance, we have to make entry in the concerned logbook. The logbook should be reviewed by the reviewer in proper time. If suppose our daily activities finish at 5 o'clock, and somebody weighs the raw material the time of exit 450 or 455. He forgot to make entry in the logbook. In the morning, next day morning, when he enters in company, what is it? The people who has arrived in the company before that, he may have made the entry of that day. So he may have escaped the entry. If the entry escaped, then you have to raise the incident or other thing, and you have to submit the document in proper way. So documentation should be in proper way. The below I have quoted use document good documentation practice GDP. Good document practice should be done. Should be there. So everybody should aware of how the documentation are made in the company in various departments. Record must be readily available. If suppose our HPLC is there, GC is there, or the other data which are electronic data, it should be available very easily. If suppose I am working on Sate sir's computer, Sate sir computer may have problem anytime because it's a machine. It is not that we have brought a new machine. It will not make any damage. It will work throughout the day. It may have some problem within the working period. So it should be stored in the proper either on the hard disk, external hard disk, or the another PC, and it should be available easily. It should be archived easily. If the auditor wants to see the records of chromatograph of other data, it should be easily available. You cannot say that, okay, sir, we are not properly store it, so we have to have some more time to gain this record. It should not be there. Okay, the document must be permanent. They should not be use of pencil. If you use the pencil, there is the chances of we are correcting our entries. No, this should not be any pharmaceutical fee. It is a big discrepancy in the pharmaceutical fee. If you have made mistake, suppose today is 21st day, and we have signed out yesterday day 20, so then we have to strike that entry. We have to sign, but which day on which day you have made a mistake, and again you have to 21/02/2021. That entry should be made. It should be legal and legible and clear. Everybody should have proper handwriting. We should not override any entry. It should be accurate also, and it should be timely completed. That I have covered 5 minutes ago. It should be timely completed. Whenever you are doing the activity, you have to make entry in the concern logbook or related computer. A sample should be retained. Each raw material and each finished product should be retained. Retained means if suppose we require, it's a 500 kg batch is raw material came in company, we require 30 gram of sample for the testing purpose for QC paper for QC. Then we are sampling the raw material in the quantity 35 gram or 40 gram, and for the 10 gram or 5 gram additional sample, we keeping it as a retention sample. If some discrepancy is happen in future, if some market company raise, then again we have to go for the retesting of raw material. That is why we are retaining the sample. Similar way, finished product is also retained by the QA team. So retention of samples should be there. It is not that we have sampled the 10 tubes, we are destroying the samples, and we have not that's my quantity. So then again we have to raise the very incident or that, and again we have to go for the sampling in production. It should not be there. Stability study. Establish the length of time in which the product meets all specifications. If our specification limit is 90 to 120, suppose our specification limit, the drug which contains should have the potency, should have the assay limit of 90 to 120. Our product should give 90 to 20% the result throughout the expiry date. If it is less, as I said, it may be OS. or it may be OT. Suppose our results are we are manufacturing 10 bag. Now for 10 bag, we are getting result 95 to 110, 105%. And if new person came, he may have made mistake during the analysis, and if the result comes 90%, then there is a OOT out of 10. The trend is 95 to 105. No, the trend he has overcome the 10. The result are 90%. Then we have to raise the OOT. We have to investigate. A senior person does the analysis, or he or her, he or she can analyze it again, and then we have to prove the batch is really get no, it is not that much effective. Then we have to intimate the production people. Then may have made some formation change or other changes which are required for the stability of the batch. Huh? Monitor the drug for this period of time. That means the monitoring of drug should be there on every period. 6 months, the FI should not be changed. If it is described the white homogeneous stem, it should be white. It should not be off-white, or it shouldn't be like another way. It should be white. Huh? pH limit, if the pH limit is 4.5 to 7, the pH should be above 4.5. It should not be less than pH 4.5. So we should do the monitoring of drug by that time. Now the sale, no distributor or importer shall sell a drug unless it has been fabricated, packaged, labeled, tested, and stored properly. If suppose gentamicin injection is there, it should be stored. I don't know the exact temperature. Suppose it has to store for 0 to 200, 0 to 2 degrees centigrade. If it is not stored properly, if the company, some people, there are different type of people in company. Directly sell, he may have escaped all the checking, and he have escaped all the testing, and he may sell the medicine or the injection or the ampoules outside he carried out outside the premises. And if it is sale, if it is not stored within that temperature, it may be harmful for the body. It may be harmful for the health of the customer. So every drug which is sold outside should be fabricated, packaged, labeled properly. It should be tested and stored properly before it goes to customer. Why GMP is important? A poor quality medicine may contain toxic substances that may have unintentionally added. It is not the intentionally somebody is adding some toxic substances within that formulation. It may create. In quality control, we are doing some test, residual solvent or OVI. What is that? It may be due to the reaction within the drug which creates residual solvent. So we have to make our medicine free from the toxic substances. The medicine that contains little or none of the claimed ingredients will not have intended therapeutic use. If suppose we are manufacturing the multivitamin injection, which is useful for our sister when they are pregnant, multivitamin, the folic acid is there, vitamin B6, vitamin B12, cyanocobalamin, other drugs are there. If it is lagging one of the drugs, the intended therapeutic effect is not there. It is we are making very big offense against the customer, so it should not be there. Some of the main risks in the good manufacturing practice, unexpected contamination of products causing damage to health or even death. In the last 1 year, we have seen during the curing of this corona patient, some deaths are happened in overall in the world. So how this has happened? There should not, the precautions are not proper. It is not it may be one reason, but the precaution should be there. There should not be any type of contamination. Incorrect labels or containers, which could mean that the patient received the wrong medicine. Yes, suppose Occupal, our product Occupal D, Occupal D. These are our products. If doctor prescribed Occupal D, and that owner of pharmacy gives Occupal DA, it has some additional drug. which is not referred to the patient, which is the not required for the patient. And if it takes occupied D, it may be harmful for that. So label should be proper. Sometimes what happened in my previous company when I was working, what happened? We are manufacturing our liquid. We were manufacturing liquid injection. We are filling it in 30 ml vials. 30 ml vials. When I went for the second shift, the second the production people, the QA person, we went for the second shift. No, in meanwhile, I think about it was about 7 p.m. We see that instead of vials, no, VILAS, it was villas, VILAS, and that is printed on about 20,000 vials. So we have to delabel that all the vials. The extra work is going on. The action is taken again. The supervisor who are work that in the morning shift and the whatever. This should not be happen. The labeling should be proper. Huh? Insufficient or too much active ingredient resulting in ineffective treatment or adverse effect. Insufficient. Insufficient drugs should not be added. There should be proper quantity of ingredient in the manufacturing, which should not make any adverse effect. And just I want to conclude the pharma industry is regulated by GMP. Every pharma industry is regulated by GMP. Now the CGMP scenario is there. Current good manufacturing. That may be, if we have a time, we may discuss later on. And good manufacturing practice must be followed each one of person who are working there. Huh? GMP ensures drug products are safe, pure, and effective. GMP ensures drug product is safe. Our motive is should be our drug should be safe, pure, and effective. It should not be harmful for anybody who is the beneficiary of the drug. Huh? It is our main effective, main motto. Huh? So here is my, I have concluding my JMP. If anybody want to have question, anybody can ask. If I am unable to, the guard sir is there, Kami Sayar, more senior than me, has more knowledge than me. He can also participate. I don't. So, Gautam sir, my
Yes,
talk
sir.
is over. Anybody
Yeah.
want to ask? Anybody have the question on the YouTube? No, I
Yes,
can.
sir.
If
Yes,
I
sir.
am able, I will give. Otherwise, certain guard sir will also answer him.
Yes, sir. Yes, sir. Sir, thank you so much, sir, for your valuable, nice presentation and nice lecture, particularly on the GLP, good laboratory practices, GMP, good manufacturing practices, GDP, good documentation procedures, practices are there, expiry of the drug, importance, and some factors related with the particular risk of contaminations are there, and some labeling. So it is a very nice, miss, we can say what that particular lecture, and I hope our students, yes, they got clear about the particular some concepts which is mentioned in our curriculum, and hopefully they will get the more clarification about by just attending this lecture. So I hope everybody stood under, miss, they have understood the particular the factors which is related with that what you have covered today. So thank you so much, sir. Thank you for your, yes, this nice lecture, nice presentation. Yes, I would like to ask students, especially I would like to, yes, anybody have any problem or any queries, especially any doubts regarding this GLP, GDP, GMP, be frank, and yes, feel free, you can ask any questions, query related with this. So this is a question answer session. So yes, any students, suppose you're having any queries, so you can write in the chat box, otherwise you can ask. Yeah, students, yes, those who are attending this particular lecture. Yeah, hello. Yes, anybody have any doubts, any queries regarding this? If you are not able to ask, then you can write in the chat box also. Even those who are attending through the YouTube channel, yes, you can post your questions, queries into the chat box. Yes, anybody else? Any students from third year, final year, even many of the students are attending from the diploma, second year diploma students. Yes, anyone else? Hello? Yes, anybody have any queries regarding this? Yes, Mr. Vinay Gavde. Miss Vinaya Gavde, 21. Do you have any query regarding this? Yes, any students from third year? I think no one has any queries regarding this. I hope everybody has cleared their concepts regarding this GMP, GDP, GLP, and some other factors related with the drugs manufacturing.
Okay.
So now moving towards the, I would like to request our principal,
Nay,
Dr. V. Yes, sir.
hello, Gautam sir.
Sir.
Sorry
Yes,
to
sir.
interrupt you.
Huh.
Due to electricity issue, sir has been in offline mode for few
Okay.
minutes.
Yes,
Okay.
sir. Yes, sir.
Once the problem is get solved, sir will going to join us. Okay.
Yes, sir. Yes, sir.
So
Okay. Okay.
we can move forward for next
Next
session.
lecture.
Okay.
Veer
With
Dhaval,
our
sir. Yes, sir.
lectures. Okay.
Yeah. Okay. So now moving towards the second industry expert lecture, Honorable Mr. Veer Dhaval Kamble, sir. He is a deputy manager at Glenmark Pharmaceuticals, Kolwale, Goa. So before proceeding this, I would like to introduce about Honorable Mr. Veer Dhaval Kamble, sir. Mr. Veer Dhaval Kamble, sir, he is a deputy manager at Quality Assurance Department, Glenmark Pharmaceuticals, Goa. He has done his Bachelor of Science in the 2003 from Swami Ramananda Tirth Marathwada University, Nanded, Maharashtra. He has done his Master of Science in the year of 2005 from Swami Ramananda Tirth Marathwada University, Nanded. Sir has a Honorable Mr. Veer Dhaval Kamble, sir. He has a total 15 years experience in quality control department with reputed pharmaceutical industry like Glenmark Pharmaceuticals, Watson Pharma, and Lupin Pharmaceuticals. Uh, Sir has worked as a group leader for non-routine raw material finished products in process instrumentation and stability sections. Also, Kamade sir has worked as a subject matter expert SME for all QC systems in regulatory audits like WHO, US FDA, UK MHRA, KFDA, Russia, and so on. He is also worked as a safety caretaker and firefighter. Previously, Honorable Mr. Kamble sir, he had worked at Lupin Pharmaceuticals Limited, Tarapur, Maharashtra since 2006 to July 2010. Mr. Kamble sir, he had worked at Watson Pharma Limited, Varna, Goa as a executive in quality control department since July 2010 to May 2013 in quality control department. And presently, Mr. Veer Dhaval Kamble sir, he is working in Glenmark as a deputy manager in quality assurance department. He is handling the responsibilities for overall laboratory compliance, responsible for stability studies for commercial and exhibit the we can say what commercial validation products, and responsible for lab supports and other. So this is just brief introduction about the our today's guest industry expert lecture, Mr. Veer Dhaval Kamble sir. Sir, over to you, sir. Please proceed for your hello. So I would
Sir.
like to request Honorable Mr. Veer Dhaval Kamble sir,
Okay.
proceed for your session.
Hello, Gautam sir.
Yes, sir.
I'm sorry to interrupt you. Please give
Yeah. Yes, sir. Yes, sir.
sharing
Just yeah.
authority
Just I'm
to sir.
giving. Yeah. Yes, sir. Please.
Yeah. So shall we start?
Yes, sir. Please, sir. Proceed. Yeah.
Sir, you
Please.
can share your PPT, yeah, sir.
Yes, sir.
Virdhar, sir.
Yeah, I'll do that. Yeah, I'll
Yeah.
do that.
Yes, sir. Please.
So first of all, thank you so much for such a beautiful introduction. And again, I just to introduce myself, Veer Dhaval, currently working as Deputy General Manager in Glenmark, and I have a 15 years of experience overall in quality control and currently in quality assurance also. So first of all, I'm audible to everyone.
Yes, sir.
Yes, sir. Yes, sir.
Yes, sir.
You're
Okay.
audible.
I like I'm very much thankful to entire Yashwantrao Bhosle pharmacy team, the faculty who has given a chance to present myself.
Yes.
And the entire students, I warm welcome for this presentation, which will be on overview of pharmaceutical industry. We will be covering how the pharmaceutical industry works, specifically a particular unit, and I'll try to make sense, by explaining the things. And a very, a great congratulations to all of you who have chosen this pharmacy field, because you all must be known, like last year was a pandemic and was a -1 year for all of us. Again, which has given multiple things, like we come to know the safest place in the world, the planet Earth, is our home, and the nearest people who are with us are our family and other team members. And also, essentially, the essential things for a living being are only two. One is food, which is produced by our respected farmers, and second, very important, is our medical field. So medical things, and in which we played a very important role. Unless we manufacture the drugs, it will not be possible to treat a patient. So it's very good that you have chosen this field, and let us start this presentation. I'll just share my PPT. You can see PPT.
Yes, sir. Yes, sir.
Yes, sir.
Yes, sir. Thank you.
Okay. So, an overview of pharmaceutical industry, how a pharmaceutical industry works. Based on the limitations of working on a having a presentation on virtual background, there will be a limitation, multiple limitations. But if anyone has any question in between, maybe students, maybe faculties or other speakers also, you can interrupt me and we can have a discussion on that. Okay. So first of all, what is a drug? So drug is a chemical substance that interacts with the living system and produces a biological response. So this is a basic definition of drug, what we manufacture. So now these drugs, specifically synthesized and biodrugs. There are two types of drugs. One are synthesized, which are chemically synthesized, and second are biological drugs. So any chemical entity, when enters inside the living being, enters inside the living being and reacts, and gives some outcome which we call it as a treatment. Okay. Second, in this entire presentation, we will be discussing the different functions which works in the industry and their responsibilities. Okay. So as a whole, pharma industry is specifically categorized in two different parts. One is a manufacturing, and second is a CRO. CRO is nothing but the R&D. So there are multiple other sections also, but the major category is of two. One is production and second is R&D. So if you see, now if we discuss about the manufacturing unit, which is again called as a pharma production house or a manufacturing site, and you can say the name. It depends on the different organizations. So it is specifically categorized in 5 different parts. One is production, which is a manufacturing unit, which is a manufacturing area, department we can say. Second is the quality control. Third, quality assurance. Fourth is pharma engineering, and fifth is regulatory affairs. So in few further slides, we will discuss one by one area and there how it works. So two important personnel in the plant are head of production and head of quality control. So now if you see the production, there are three different types of areas where it is categorized. One is formulation, second is screening, and third is packaging. So once the manufacturing is done, the product is packaged and then it is dispatched to the market. Now, what are the responsibilities of head of production? Products produced and stored according to the so specifically the production manager plays a very important role. He has to ensure the basic GMP, what our other speaker has, Sandeep has explained. So approval of production instructions, their strict implementation, ensuring of production records and evaluation and sending to QC, maintenance of department, premises and the equipment, and the validations of the instruments and the products. And it's very important is the persons who are working in the area should be trained. The quality control department is again categorized in 3 different parts. First is chemical QC, which is again API testing. Second is finished product testing, and third is instrument QC. So in quality control, basically once the API and raw materials are received, they are again analyzed, and after release, these materials are given to the manufacturing department. They produce the manufacturing using different steps, and again the sample is given back, the finished product given back to the quality control department, and then further it is analyzed, and the quality is checked against the specifications which are established for a particular drug. So for every drug, for every product, there will be different set of specifications. The second department what we have is called microbiology, where specifically microbiological testing is done. So for a particular product, there should be few organisms like Staphylococcus aureus, then Escherichia coli, and other pathogens which should not be available, which should not be present. Then they are tested and ensured that product for example, our oral solid doses, that is tablets or capsules, are not having any microbial load. Also, the environmental monitoring has to be done in the production areas wherein the manufacturing is done, or also all the critical areas of the manufacturing facility and the quality control also. Third very important department is packaging QC, where the packaging material testing is done. The artworks means the label what we have on the product is reviewed, verified for each and every minute detail, and then pack insert checks are again reviewed. So further, the head quality control, which are the responsibilities of a head quality control. So head quality control and the entire quality team has a right to approve or reject a material. So which includes all types of components, right from packaging materials, raw materials, active ingredients, excipients, finished products, everything. Then evaluate the batch records, ensure all testing is performed, approve the specifications, ensure that if error have been occurred and it is fully investigated to identify the root cause and the CAPAs, corrective actions to avoid further recurrence, approve and monitor contract analysis, maintain department premises and equipments. So these all aspects are in detail explained by Sandeep, wherein he has explained the GMP, which is more or ICH Q7, and 21 CFR part 211, wherein he has explained all the requirements with respect to, for example, when it is told like maintain department of premises and the equipment, what is to be done. He has very well explained. The next part is validations performed. Any types of validations, whether it is a software validation, whether it is a process validation, these all types of validations has to be done and ensured it is done as per the established procedures. Next, ongoing and initial trainings. So training is a very important part, because over a period of time SOPs are changed, the guidelines are introduced, the guidelines are updated, the requirements are changed. That changes has to be done, and again everyone in the system has to be trained. So shared duties of head production and QC, again they both are having a common few things which are common. That is authorization of written procedures and other documents, monitoring and control of manufacturing environment, plant hygiene, process validation. Now process validation, I'll just explain in brief. Process validation, whenever a new product is introduced, all the critical process parameters of a manufacturing process, whether it is a sifting, milling, compression, packing, everything has to be reviewed for the minimum and the maximum. So we have to set a range. So it is always not possible to have a product at 100%, for example. It may have sometimes 102 also, it may have sometimes 96% also. So based on the process validation, we need to decide the range of the entire manufacturing critical process parameters. Then training, approval and monitoring of suppliers. The suppliers in the form of, for packaging material suppliers, the excipient suppliers, the raw material suppliers, and so on and so forth. Approval and monitoring of contract manufacturers, designation and monitoring of storage conditions, retention of records, monitoring of compliance to GMP, inspection, investigation, and taking samples. So these are some shared responsibilities of aid production and quality control. Now, third very important department is quality assurance. So quality assurance, you can say, is a department who takes care of, it's a backbone of the entire pharmaceutical industry, wherein all the departments are directly monitored and guided to follow the GMP by quality assurance department. So quality assurance takes care of everything, right from your receipt of material to release and dispatch in the market also. So quality control, quality assurance department has two parts. One is system QA and second is process QA. Now, what does system QA does? System QA does the vendor audits. So vendors means the suppliers who supplies the excipients, APIs, or other services like calibrations, qualifications, validations, and so on and so forth. Second important part is your internal audits. So we have, like, QA has to do the periodic internal audits, wherein they have to check the systems which are written, the procedures which are written, the SOPs which are written are being followed consistently. Third is change control. So change control is a device or a mechanism wherein any type of change in the existing system, whatever it may be, like small, large, major, minor, critical, whatever, any change has to be first initiated and given to QA for approval. So once it is approved, the change has to be implemented. So I say koi change liya hum logon ne and then it has to be implemented is not possible in pharmaceutical industry. Then CAPAs, in case of any failures, the corrective actions and the preventive actions to avoid the recurrence and to correct the error. The complaint handling. So during the course of manufacturing, if something goes wrong, or the transportation, if something goes wrong, and the product shows some, error. For example, a basic error is change in the appearance, or change in the color, or a discoloration, any sort of issue, or a lack of effect. For example, if I am taking a product which is for my headache and it's not working, so it's again a complaint. So these complaints are received at the site and to the industrial unit, and that is taken care by QA, whether that complaint is actual complaint or it's a wrong interpretation of the customer. The last is training. QA has to ensure the entire plant is within the training status, and whatever changes implemented, that has been gone through a rigorous training mechanism. Second QA, what we have is a process QA, which is specifically involved in the IPQA. So which is IPQA, when it is said IPQA is the in-process quality assurance. So in-process quality assurance is a department, a section, a group of people which is involved in the assurance to throughout the manufacturing process. So all the steps of manufacturing process are commenced as per the written down procedures, which is called as a BPR or BMR, like batch production record or batch manufacturing record. Second is risk assessment. In case of any changes, the first assessment has to be performed, whether this change will have any impact on the product, on the product identity, quality, integrity, and efficacy. Third is deviation. Now, in case of any procedure which is written, and in any event that has to be deviated due to some constraints, for example, in pandemic we have few deviations because due to the lack of resources and sort of. So that deviations has to be first initiated. A risk assessment has to be done. Once it is been approved, then that has to be implemented. Next is OS and OT. So out of specification, out of trend. So name itself indicates these are the departures of the product quality from the written down specifications, from the written down range. For example, if I have a specification for assay 90.0 to 110.0%, and if the product shows assay below 90 or above 110, then that has to be investigated to identify the root cause. Second part, process validation, as we have explained, equipment validation. So whatever equipment which are used in the laboratory or in the manufacturing process, that has to be verified for its intended purpose. And the software validation, now software validation is a thing which is now has become a disaster for entire pharmaceutical industry. Not only in India, it is globally, because as you might be aware, regulatory authorities like US FDA or MHRA or MCC and so on and so forth, they are reviewing the electronic data, and what they are finding, it is down the line 5 years back. Now also in couple of companies it has been observed. So things are not, the printed copy which is there is not in line with your soft copy. So we need to ensure these software validations are done, and there should not be deletions of the data, the updation of data. Each and every user should have a specific ID, so that who has done the activity, that is to be captured. So as we have discussed, role of QA, specific responsibilities, control of licenses. So for manufacturing and distribution of a product, of a drug, one has to obtain a license. For example, in India, our local FDA provides a license. In case of US FDA provides a license, and it's differed for individual country. So liaison with R&D and the new product introduction. It's a communication bridge between the R&D and a new product at site. So batch review, batch release, management of the document management system, management of the internal audit, management of GMP training systems, customer feedback and the satisfaction, management of deviations, incidents, investigations, management of validation in the system, and vendor assurance and supplier auditing. So these are the basic roles of QA. So engineering department, pharmaceutical engineering department, again a qualification of equipments, qualification of whatever equipments which are used in the manufacturing, quality control and engineering department. Then FAT, SAT. FAT is a factory acceptance test, wherein a particular equipment is asked, and a QA person has to visit that particular manufacturer, and he has to verify the instrument is prepared or manufactured as per our requirement. IQ, OQ, PQ, DQ, these are the qualifications. IQ is installation qualification, operational qualification, performance qualification, and design qualification. So these are it's a huge terminologies, and it is very much important and very much elaborative. So we'll just keep and let's proceed further. And the maintenance team. So maintenance is a to understand, to maintain the consistency of the equipment, a periodic maintenance has to be done. Otherwise, what will happen if periodically wear and tear parts are not replaced? The equipment may land up in the malfunction or activities about it. So it consists preventive maintenance, calibration, and breakdown maintenance. So specifically in the manufacturing of medicines, equipment use, this we have discussed actually in previous slide. So the head of engineering has basic roles like maintenance of premises and equipment, design of facilities, so how the facility should be, validation and qualification of equipment and facilities, management of the calibration program, management of the planned preventive maintenance program, control and operation of site services, including water, steam, and HVAC systems. Now these HVAC systems are basically air handling units to ensure the cooling in the at the defined temperature. So that system, and like routinely in the offices or at home we use the split ACs, but in the industry, like pharmaceutical industry, the HVACs are used, which are the centralized cooling units, wherein the different filters are there to avoid the entry of larger particles and the microbes inside the areas. So the maintenance of that part. then control of engineering spares and equipment spares. So, like I'm not too much fast now. The space is okay. Like,
Yes, sir. It's okay, sir.
yeah. Otherwise, मैं बोलते जा रहा हूँ, then ऐसा नहीं होना चाहिए
No,
कि
no.
like,
Yes. Even suppose
okay.
some students may have any queries, you can ask me, sir.
Yeah,
Okay.
in
Other we
between.
will have yes at the end question and answer session, sir. No problem.
Also, if someone has any particular question on the slide, you can raise your hand. The facility is there in Zoom platform also, and we can have a chat. Okay. So further, the head of manufacturing has these roles: control of engineering contractors, approval of engineering related procedures, documents and drawings, control and storage of engineering records, control of equipment manuals, pest control to avoid the rodents, and the flies and so on and so forth in the areas in the entire manufacturing unit, site security and access, and the project management. So these are the basic roles of head of manufacturing. So quality management system, it is specifically to handle the quality complaints. So pharmaceutical quality management system, that is PQMS. FDA has a guidance which is called as guidance for quality systems. ICH has a specific guideline for that. That is Q10, which is specifically guidance on pharmaceutical quality systems. So these are a few set of guidelines which mandates the different procedures to be followed inside the factory. So if you go through, like ICH has about 14 guidelines, ICH Q1 to Q14. So for each and every activity, there is a set of requirements, and the facility has to ensure that requirements are fulfilled, which is periodically reviewed by the auditors or by the agency, any agency like USFDA, MHRA, I named it in previously. So they visit the site periodically and they review whether the site is following all these guidelines. And if they found satisfactory, further manufacturing will be continued. If they found any kind of observation, they give an observation and the factory has to respond to that query. If suppose a regulatory agency found the site is absolutely not following the GMP, the site will be shut down. So that's how it is. The ICH Q10 model, which is specifically describes a comprehensive model for an effective pharmaceutical quality system. So this guideline, as we have seen, like pharmaceutical quality system, it talks about pharmaceutical quality system, how the quality system should be there on the site. Then there are other guidelines like ISO, then good manufacturing practices, then ICH Q8 for pharmaceutical development, Q9 for quality risk management. So I totally understand. When I was in college, maybe on 11th, 12th, and up to BSc, MSc, these are all terminologies we were not aware of. So when we have started inside the in industry, after 3, 4, 5 years, we understand these guidelines. As I'm telling you, it is quite possible you will not understand it now, but at least you will come to know where we have to review, like what requirements are there. You can visit these sites. So there is a guideline, like there is a site called ich.org. You can visit that site. You will find, it is freely available to everyone. You can download the guideline, you can read it. So that's what I'm telling you. It is very difficult to understand in initial stages, but since the requirements you will come to know, it will be better for you to resource in case of any queries. So QMS enhance the quality and availability of medicines. Okay? It always facilitate innovation, continual improvement, and strengthen the link between pharmaceutical development and manufacturing activities. So, sorry. I'm sorry, I just lost it. Okay. So pharmaceutical development has few things like drug substance development, formulation development, manufacture of investigational products, delivery system, manufacturing process development, and scale-up batches. Scale-up batch is a trial batch, you can say. When the formulation develops a particular product, they first create one batch which is not sent in the market. That will be just for understand whether we have manufactured a good product, and if a particular factory found it is satisfactory based on the stability and the tasting of that drug product, then further it is started with stability and then plan of distribution inside the market. So technology transfer. So if first of all, like a product is developed by the FR&D is a formulation R&D at a different, like which we have seen as a CRO. And once the product is developed, that product is transferred inside the manufacturing facility, and that transfer is called as a technology transfer. So new product transfers during development through manufacturing, transfers within or between manufacturing and testing sites for marketed products. Now the commercial manufacturing. So commercial manufacturing is a process where we manufacture the drug, pack it, and directly send to the customer, like distributed inside the market. So acquisition and control of materials, provision of facilities, utilities and equipment, production, that is packaging and labeling, quality control and assurance, release of the batches, storage of the batches, and distribution inside the market. So product discontinuation in case of any failures, in case of any problems or any quality complaints, the retention of documentation, sample retention, and continued product assessment and reporting. So these are the few things. So as we have seen, quality risk management, ICH Q9, which is specifically for the risk assessment. For why it is there? To identify and scientifically evaluate and control potential risks to the product quality. So at the same time, there are two, as we have discussed, there are two important factors inside the pharmaceutical industry. One is CRO, that is R&D, and second is pharmaceutical unit or site. Apart from these, there is one separate team which is working, which is called as specifically corporate office. Corporate office takes care of multiple things. So this particular part is the management's responsibility. So what is the management responsibility? Senior management ensure an effective quality objectives. So management should be focused to produce and ensure effective quality objectives. They have to ensure the quality policy is in place and it is been followed. So quality policy is that describes the overall intentions and direction of the company related to quality, an expectation to comply with applicable regulatory requirements and continual improvement. Policy should be reviewed periodically for continuing effectiveness. So these are the quality policy requirement. Third is quality planning. So quality objectives should align with the company strategies and be consistent with the quality policy. Then management should provide the appropriate resources and the training to enable the maintenance of quality at all times, consistently maintaining a quality standards. Further, they have to ensure resource management. Resource management is nothing but the manpower which is working, the equipments, and other stationery and sort of like all other things. Internal communication in case of any changes, in case of any upgradations, that has to be communicated to the entire team. And the management review. Management review is a central process wherein the entire manufacturing unit is reviewed for all types of changes, for all types of failures, for all types of outcomes, and all types of errors. Quality assurance, as we have seen, like there are pharmaceutical law, good manufacturing practices, good laboratory practices. This part already covered by Sandeep. Good quality assurance systems leads to a constant increase of reliability and ensures compliance with the current regulatory requirements. Now there is one other department which is very important, which works as a bridge between a regulatory agency, that is local FDA. For India it is a FDA, for US it is a US FDA, for Europe it is a UK MHRA, and so on and so forth. So between that particular regulatory agency and the pharmaceutical industry, so there is one department who takes care of that, who works in between, like information is taken from site pharmaceutical industry to the agency and vice versa. So what this regulatory affairs department works is pharma products regulated markets, US, Europe, MCC, and to filing of DMF, the filing of CTDs, ECTDs, and NDA, ANDS. So NDA is new drug applications, ANDA is abbreviated new drug applications. Now this ANDA is a generic products, what we can see. So there are two types of drugs which are manufactured. One is a patented drug, which is first time discovery by a particular company. So if a particular company produces a specific drug, there will be manufacturing, which is called as the NDA, and for next 20 years, nobody can manufacture that because it is patented product. After 20 years, that drug is released for anyone can manufacture that, any company can manufacture, and that is called as a generic market, generic drugs. And in the term of filing, it is called as a ANDA, so abbreviated new drug applications. So other stream is there, which is biological and biotech products, and markets are regulated and semi-regulated also, rest of world other than US, Europe, and the filings are DMF, open part, closed part, resignation, registration, dossier, and take backs. So these are different terminologies using which an entire manufacturing process, along with the quality of standard, is submitted to an agency. Once it is submitted to agency reviews that, visits the site, ensure everything is followed as per GMP, and then approval is given, the license is given to manufacture and distribute inside a particular market. Responsibility for managing the interface between the sponsor regulatory agency and the site, as this we have discussed. Submission to regulatory authorities. So what we have seen is dossiers, then NDA, ANDA, then take packs and all this. The name it changes country wise. So that entire package is submitted to the authority, regulatory authority, Europe, US, Indian FDA, and so on and so forth. And the work, that particular activity is done by regulatory affairs. So participates in supporting and promoting electronic submissions and electronic archives. Ensure that regulatory documents comply with the relevant guidelines. Again, they have to verify the take back which is received is in line with the regulatory guidelines. Identifies and records issues that require resolution prior to finalization, and the bridge between the responsible author and resolves any issue. Assist authors in case for the completion and compilation of regulatory documents to ensure all components are provided and presented in the correct format. May provide training based on the requirement. So clinical research is a R&D. So the process is, if you can say, first is clinical operation, means a batch is a particular product is developed. Pharmacovigilance, this reviews the entire take pack with respect to safety, medical monitoring, data analysis, and ADR reports. Third is biostatistics, that is validation of product, SAS program, lead data management. Next is medical affairs. Here that particular product and the outcome is discussed with the medical advisors. Then further it goes to QA/QC, where testing, facility audit, training, and GCP compliance. And finally, regulatory affairs. The entire take pack, which is produced using all these aspects, is finally submitted to the regulatory agency for approval purpose. So these are few regulatory guidelines which are available, which are been followed. Like ISO guideline is there. There is a WHO guideline which is given by World Health Organization. I'm just giving these requirements, for your reference purpose. There is one USFDA guideline which is for United States. There is one ICH guideline which is a centralized body considering all countries. Then PICS guidelines. There is one IPEQ guidelines. So these are different guidelines which will give you an insight how the industry works. UK MHRA for Europe, we have seen. South Africa, MCC, Medicine Control Council. Australia has a TGA, that is Therapeutic Goods Administration. India, as we have seen, Drug and Cosmetic Act, and also known as the Schedule M. Schedule M is a good manufacturing practices. So in Indian pharmacopoeia, as per Indian FDA, IFDA, there are different schedules. So these schedules give a specific requirement of a particular process. For example, Schedule M talks about good manufacturing practices. Schedule U talks about good documentation practices, and so on and so forth. So like EDQM for European Directorate of Quality and Medicines and Healthcare, New Zealand has a MedSafe, Gulf has GCC, you know, Japan PDMA. So these are different countries with their regulatory authorities. So nowadays, a very important factor which is there inside the industry, pharmaceutical industry, and wherein many 483s and warning letters and plants are shut down because of this requirement, which is called as a data integrity. So data integrity is a protection of data from unauthorized and unaccountable changes. Now, how this data integrity has to be ensured? It is ensured through a ALCOA principle. Now, ALCOA is an acronym of A stands for attributable. Attributable is data should clearly demonstrate who observed and recorded it, when it was observed and recorded, and who is about, like, who has done it specifically. L stands for legible, means data should be clearly legible in a readable and printable format. Contemporaneous means the activity, whenever it is commenced, that has to be immediately captured, and the records has to be ensured. ऐसा नहीं होना चाहिए कि like once the activity is done and the reporting is done after 2 days, so that will have a bias in your data. Fourth one is original. Sorry, data should be original, Xerox वगैरह नहीं होना चाहिए. And the fifth is accurate. Data should be error free and conform to the protocol requirements. I'm sorry. So how it can be done? One is audit trail. Second is 21 CFR Part 11. This is a US guideline for electronic records and electronic data. Third is computer system validation. Fourth is secure documents. Fifth is backup and recovery. Sixth, user training. And seventh is internal audits, periodically review of the systems. So what is next? So, like, as we are in the student stage, build your knowledge, read the things, read all the sites which we have, written. Now the information is on the, the one, it is on fingertip now. You visit any site and you will get the knowledge. And what to do? Knowledge, skills, abilities. Increase your knowledge, not only with respect to industry, with respect to whatever you do and whatever you have planned to do in future. Improve your communication skills. It is very, very, very important. Once it has been asked to Warren Buffett, like you might be aware, Warren Buffett. Who is Warren Buffett? Warren Buffett is a man who is, one he is the rich, and second he is a philosopher. He is one of the most successful guys now with our era. Once it is asked to him how one can increase his value, and the answer he has given is one has to ensure he has a robust communication skills, oral as well as in the writing. So once you do this, your value will be increased by 50%. So we need to strive to improve the communication skills with respect to written also and with respect to verbal also. Since we are in a student stage, we can do this, and we need to do that. Third is reasoning ability. See that how we can improve our problem solving ability. Then the fourth is leadership. So try to improve your leadership qualities. There are multiple books available, multiple seminars available on virtual platform also, wherein you can attend that, where you can, based on that information, you can improve your knowledge and the leadership qualities also. So that's it from my side. Any questions? If have, we can discuss in detail.
Yes, thank you so much, sir, for your nice session about this quality assurance department system like that, NDA, ANDA, DMF, tech pack, qualification and maintenance of the team, IQ, OQ, PQ, DQ, so on. It is a compact package for the, especially those who are the learner, for especially those who want to pursue particular, we can say what, to join the pharmaceutical industry. So this is a compact package is there. And I hope students, those who are right now attending this webinar, or we can say this particular session, so that will be a, definitely it will be a fruitful to especially our students. So yes, thank you so much, sir. Nice presentation, and nice, you have nicely delivered this nice, we can say what, particular this topic. And yes, I hope our students, they got understood the particular concepts about this quality and assurance. So yes, anybody have any queries, especially, so I would like to, yes, those who are having any queries, any doubts regarding this, you can ask to Honorable Mr. Virdaval Kamde, sir. Yes, please. Anyone? Yes, anyone? Yes, those who are attending this particular webinar, or we can say this particular lecture session. Yes, anybody have any queries, so you can. Hello.
See, as I, I'm sorry,
Yes, sir.
as I
Yes, sir.
told
Please.
you, in our college days, I'll take my own example. It was very difficult to understand all these terminologies, but yes, one should know where we have to go. So this is just a brief idea. What are the things which are there, and we can pursue further in detail knowledge of that. Each and every concept, each and every terminology has a huge knowledge, huge literature. So now also, if someone is not having any questions, I'm reachable, and we can have a discussion on that.
Yes.
So at the end, I would like to thank you, Mr. Vijay Jagtap. With the help of my another speaker, Shrikant Garg, my colleague, like he has given a chance to discuss with such a wonderful audience. It would have been great to visit a college, but
Yes, sir.
unfortunately we
Sure.
could not.
Yes.
And I am very much thankful to you and your entire technical IT team who is behind this. This is not a simple task. I know that there are multiple things which has to be taken care, and that's it. Thank you so much.
Yes, sir. Thank you so much, sir, for your nice presentation. And yes, nice, Miss, we can say this is topic, and definitely it will be helpful to bridge the gap in between the industry as well as the academia. Thank you so much, sir. Thank you for your valuable guidance to our students. Now moving further, moving towards the last industry expert lecture. Today we here with us Honorable Mr. Srikant Gad, sir, Quality Assurance Executive, Glenmark Pharmaceutical Limited, Kolwale, Goa. So before proceeding this particular last session industry expert lecture, I would like to introduce in briefly about Honorable Mr. Srikant Gad, sir. Srikant Gasa, he has done his master's in science, Master of Science in Chemistry from University of Mumbai. He has engaged in the training and training of the freshers as well as giving them full industry exposure. Now, Honorable Mr. Srikant Gad, sir, he is working, or we can say it is a handling and analysis of the stability studies samples. He is also handling the chromatographic apex integration, all these particular related with the quality assurance department. So right now, Honorable Mr. Srikant Gad, sir, he is working at Glenmark Pharmaceutical Limited since May 2011. So yes, over to you, sir. Yes, please, you can proceed for your session.
Okay. Can you hear me?
Yes, sir. Yes, sir.
Yes, sir. Okay. I want
Yes.
to share my
Yes, sir.
PPT.
Yes, sir. I enable you, sir. Yeah. Yes, you can.
Okay. Good afternoon, everyone. I'm Srikant Gad, working in Glenmark Pharmaceutical as a quality control executive. And first of all, I am introduction about pharmaceutical. Pharmaceutical plays very important role in our daily routine life because we are making a life-saving drug. Life-saving drugs, so it is very important and very important roles in human beings. So pharmaceutical industry also from last year, pandemic year, we are plays important role about essential supply of drugs. So pharmaceutical very and my today's topic is instrument used in pharmaceutical quality control laboratory. Okay. So I'm going to discuss all these topics: disintegration, HPLC, then dissolution. And first, my first instrument. This is the instrument plays important role in quality control. First instrument is a disintegration tester. Disintegration tester means it is time required for the dosage form break into granule specified size और under careful specified condition. इसका मतलब है कि यहाँ पे आप इंस्ट्रूमेंट देख सकते हैं. इंस्ट्रूमेंट में वहाँ पे वाटर बाथ होता है, बाद में 2 beakers are there, baskets are there. Because water bath temperature is 37 degree, because our body temperature is 37 degree. Also there are 2 baskets are there. In baskets 6 open-ended transfer tube about 75 to 80 mm long, having inside diameter about 23 mm and wall 1 to 28 mm thick, and tube are held in vertical position by 2 plates, each of the 80 to 92 mm diameter and 5 to 8.5 in thickness. So you have to put the tablets in all that 6 tablets, and you have to measure the time required to sample breaks into particles. Sample means your tablets, capsules are there, which is breaks in particle. That means disintegration time. In other words, disintegration is a measuring the breakdown of
Sorry, sir. Sorry for interruption. I am interrupting you. Better way, sir. You have to slide show. You can just begin your slide show. That will be
Okay.
more easier to see your slides. Yes, sir. Please. Yes, sir. Yes. Thank you so much, sir. Yeah.
So in other words, disintegration time is a measure of the measuring the breakdown of the dosage form. Dosage form means capsules, tablets are there, and dissolution is the measuring is the drug being solubilized in the media. So disintegration is a very important instrument, because we are measure the disintegration time, means when you take the tablets capsule, in which time, how much time required to disintegrate the tablet in our body. So see the diagram of the disintegration tester. So water level for beaker is when your basket is in upper position, then your capsules or tablet completely dipped in that water. So that completely dip in that water, and time required for that disintegrate all particles is there a disintegration time. And also discs are used for this disintegration. Disc means where some tablets capsule are floating on the media. So in that case we have to use the disc. And in this table, types of capsules for uncoated tablet, disintegration time is 15 minutes. For coated tablets is 30 minutes, and other coated tablet is 60 minutes. Enteric coated tablets. Enteric coated tablets means which dissolve in intense time, should not disintegrate in 120 minutes in 0.1 molar hydrochloric acid, and then after mix in phosphate buffer pH 6.8, that is not more than 60 minutes. Dispersible and soluble tablets, which are disintegrate time not more than 3 minutes, and for effervescent tablet it is 5 minutes. Same table is there for disintegrate time, condition and interpretation. Condition for uncoated tablet, medium use is waters or specified is monograph. Monograph is means your STP, standard test procedure, and temperature is 37 degree plus or minus 2 degree, and limit is as per your standard test procedure. And then coated tablets, it is what also water is used as a medium, and temperature is same, and limit is as per your standard test procedure. Enteric coated tablets, stimulated gastric fluid, TS, means there is a 0.1 molar HCl is used, and temperature is same, 37 degree, as per our individual monographs. And buccal tablet is a water as specified in monograph, water or specified in monograph, and temperature is 37 degree plus or minus 2 degree, and its limit is 4 hours. And for sublingual tablets, water as a medium, and limit is also as per your standard test procedure, and temperature is 37 degree plus or minus 2 degree. So disintegration test plays important role because we have to know what time required to disintegrate tablet in your human body. Okay, second instrument is used in quality control lab is HPLC, that is liquid chromatography. HPLC HPLC is a very important instrument in our lab because I think about 50% test is tested on HPLC. For example, assay by HPLC, dissolution by HPLC, then there is a related substance by HPLC, identification by HPLC. So HPLC plays very important role in pharmaceutical quality control lab. HPLC is specially based on liquid chromatography, is well-established technique for separation of substance. High performance liquid chromatography. HPLC is suitable method for the analysis of wide range of application area. Here we describe the principle of HPLC and introduce the most important component in HPLC system and the factor that determine the success of measurement. HPLC is mainly depend on separation technique. We have to separate the all ingredient from samples, and then they measure that signals and which convert in chromatogram. So HPLC mainly depends on separation techniques. In HPLC there will be three main parts is there. First is pump, second is column, and third is detector. You have to see the diagram. Pump is there in this diagram and detector is on upper side, and column compartment is there, and show the diagram for column. यहाँ पे आप देख सकते हैं कि कॉलम का diagram plays very important role in HPLC. The principle of HPLC is based on distribution. That capsules are there, tablets are there between the mobile phase and stationary phase. Stationary phase means column used for that particular analysis, and mobile phase such as organic solvents are there, waters are there, buffers are there, which is used which is mentioned in your STP or individual monograph. Depending on the chemical structure of the analyte, the molecules are retarded while passing the stationary phase. And the detection unit, for example, UV detector, recognize the analyte after leaving the column. Means for detection when signals are converted, which is detected by detector, and then signals are converted and recorded by data management system, that is software. Software particularly used in our waters. For waters SPLC we are use M4 software, and through that software we have to show the chromatogram, and then processing of that chromatogram. Also there will be checking of river, second check from quality assurance, and then you have to final the result for assay for RS. So in this detector and column, detector is main function to detect the noise signals from the column, which is separated ingredients, sample ingredients. Then there will a pump mode is two type of pump mode is there in HPLC. One is gradient and second is isocratic. Simple अगर simple भाषा में बता जाए तो gradient means there will be two mobile phase are there, and in isocratic only one mobile phase is there. In HPLC, there will be 4 ports for mobile phase A, B, C, D, and then it's one seal wash is there, then needle wash is there. So depending on the composition of mobile phase, 2 different modes are generally applicable. If the makeup of the mobile phase remains constant during the separation process, the HPLC system is defined as isocritic illusion. That means सेम वन मोबाइल पेज कांस्टेंट फॉर सेपरेशन प्रोसेस, मतलब एक ही मोबाइल पेज यूज किया पूरे एनालिसिस के लिए, तो उसे करते हैं आइसोक्रेटिक. एंड व्हेन कंपोजिशन ऑफ मोबाइल पेज इज चेंज ड्यूरिंग सेपरेशन, देन एचपीएलसी सिस्टम इज डिफाइंड एज अ ग्रेडियंट. मतलब अगर आप 2 मोबाइल पेज यूज कर रहे हैं तो उसके लिए ग्रेडियंट, पंप मोड इज ग्रेडियंट. ग्रेडियंट मीन्स देयर इज अ setup is there, how much mobile phase is changing percentage from mobile phase A, from mobile phase B. Then second important is column. In other word, column is the heart of HPLC system because it is responsible for adequate separation of the sample ingredient. अगर कॉलम अच्छा है तो आपका एनालिसिस सिंपल हो जाएगा. So for column it is washing is more important, then column storage is more important. As per जहाँ पे लिखा है कि कॉलम कैसे हैंडल करना है, कौन सा कॉलम वॉशिंग के लिए कौन सी सॉल्यूशन यूज करना है, तो वो बहुत इंपॉर्टेंट रोल है. क्योंकि अगर आपका कॉलम ठीक नहीं है तो आपका क्रोमेटोग्राम पे जो पीक आएंगे वो अलग तरह के आएंगे, बेसलाइन नॉइजी आएगी. सो कॉलम प्लेज वेरी इंपॉर्टेंट रोल. And third is pump. So pump function is transfer the mobile phase with constant flow. मतलब जो भी आपने कमांड दिया है HPLC के लिए कि 1 ml 2 ml फ्लो, तो वो उसी फ्लो में से पंप वो ट्रांसफर करेगा मोबाइल फेज. तो पंप प्लेज वेरी इंपॉर्टेंट रोल. ऑल्सो लिक्विड मोबाइल फेज अंडर द हाई प्रेशर फ्लोज. HPLC में the liquid mobile phase under the high pressure flows through an inner tube, column packed with the fine powder that is liquid stationary phase. तो कॉलम में वहाँ पे स्टेशनरी फेज रहता है, वो उसका column plays the role as separation technique which separate the samples in their ingredient. Then detector, the task of detector unit is to register the time and amount of the substance which is eluted from column. Also there is a टू नॉर्मल फेज एंड रिवर्स फेज. तो नॉर्मल फेज में क्या होता है कि हम जो भी ओनली ऑर्गेनिक सॉल्वेंट यूज, ऑर्गेनिक सॉल्वेंट मीन्स मिथेनॉल, एसीटोनाइट्राइल, एंड रिवर्स फेज, इन रिवर्स फेज वाटर इज यूज. सो इन सिंपल वर्ड, अगर नॉर्मल फेज एनालिसिस चल रहा है तो उसमें वाटर यूज करना नहीं है, ओनली ऑर्गेनिक सॉल्वेंट. और रिवर्स फेज एनालिसिस है तो उसमें आप ऑटर्स यूज करते हैं, बफर ऑटर्स. chromatographic parameters. Chromatography is very important in quality control lab because all tests such like assay, then dissolution, related substances which are only tested on HPLC. The separated analytes which are transported by the mobile phase are recorded as a signal peaks by the detector. जो भी हम सैंपल बना के इंजेक्ट करेंगे HPLC में, वो डायरेक्ट मोबाइल फेज में इंजेक्ट होके कॉलम में जाएगा, कॉलम में वहाँ पर सेपरेशन हो जाएगा और वो सेपरेट हो जाएगा. सेपरेट होने के बाद डिटेक्टर यूनिट में उसका सिग्नल रिकॉर्ड हो जाएगा. एंड द टोटल अमाउंट ऑफ पिक इज कॉल्ड क्रोमेटोग्राम. एंड वही जो सिग्नल रिकॉर्ड हो जाएगा उससे हम कहते हैं क्रोमेटोग्राम. ईच इंडिविजुअल पिक प्रोवाइड क्वालिटेटिव एंड क्वांटिटेटिव इंफॉर्मेशन अबाउट एनालाइट. मतलब जो भी पिक्स रिकॉर्ड हो जाएगा उसका एरिया, परसेंटेज, वो सब रिकॉर्ड हो जाएगा और उसका क्वालिटेटिव एंड क्वांटिटेटिव इंफॉर्मेशन ऑफ एनालाइट. देन रिटेंशन टाइम मीन्स आप जो भी सैंपल इंजेक्ट करेंगे और वो जो भी सैंपल इंजेक्ट करने के बाद वो जो सैंपल इंजेक्ट से पीक इल्यूटेड जो भी टाइम है उसे कहते हैं रिटेंशन टाइम. The retention time refers to the time which is required for a compound from the moment of injection until the moment of detection. जहाँ पे सैंपल इंजेक्ट किया वहां से पीक डिटेक्ट हुआ, वहां का जो भी टाइम है उसे कहते हैं रिटेंशन टाइम. और HPLC में so much care to be taken for mobile phase filtration is there, then sample preparation is there, because in related substance said chances to contaminate the sample. So much care to be taken during analysis, then filtration of mobile phase. You have to give follow the procedure as per written down in STP. Then third topic is dissolution. Dissolution is also plays very important role in drug substance like tablets and capsules. And in dissolution test measure the amount of drug that goes into solution over a period of time under standardized condition. Means in simple word, आपका जो भी tablet capsule है, drug release time required to drug release means उसमें जो भी API है वो release होता है, वो time मतलब dissolution time. In disintegration, we record the time to disintegrate the tablet into particle, and in dissolution, we record the time tablets API release through release in solution. And factor that affect the dissolution of the drug product include the intrinsic properties of the API. Example, solubility of the drug, then wettability, particle size, and surface area. Surface area means some coated tablets are there. And dissolution is a process by which solid substance enter into solvent yield a solution. Dissolution is dissolution भी important रोल है, क्योंकि डिसोल्यूशन में आपको पता चलता है कि ड्रग कितने टाइम में रिलीज होता है, कितना टाइम लगता है कि एक ड्रग रिलीज होने, एपीआई रिलीज होने में. सो इज अ वेरी इंपॉर्टेंट रोल. एंड इट इज dissolution, it is controlled by the affinity between solid substance and the solvent. Dissolution testing is interpretation IP standard. Dissolution testing में simple है, as per disintegration test में जैसे भी दिखाएगा वैसे ही वाटर बाद रहता है, बाद में बाउल्स रहते हैं, pedals are there, and basket is there. And also sinkers are there, Japanese sinkers are there. The dissolution testing criteria as per IP standard, quality list S1, as number of tablet tested 6. Each unit is not less than Q plus 5%. Then Q is the quantity of Q is the quantity of Q is the amount of the dissolved active ingredient specified in individual monograph. That means Q. So for example, 80Q, 90Q, which is mentioned in your STP. Then S2 criteria is average of 12 units, mean S1 plus S2 is equal to or greater than Q, and no unit is less than Q minus 15%. मतलब आप दोनों S1 plus S2 plus करना है, उसका एवरेज निकालना है, and no unit is less than Q minus 15%. फॉर एग्जाम्पल अगर आपका 80Q है, 80 minus 15% करना है, और उसमें वो no unit is less than Q minus 15%. Then S3 criteria is there. वहाँ पे 12 tablet पे टेस्टिंग करना है S3 में. Average 24 units, S1 plus S2 plus S3 is equal to or greater than Q, and not more than 2 units are less than Q minus 15%, and no unit is less than Q minus 25%. Q is the amount of dissolved active ingredient specified in the individual monograph expressed as per percentage of the label amount. तो यहाँ पे डिजोल्यूशन जब हम लगाते हैं उसमें सिंपल केयर लेना है। एक तो डिजोमीडिया बनाते वक्त उसका पीएच, क्योंकि थोड़े सम टैबलेट्स आर देयर, कैप्सूल्स आर देयर, उसमें वहाँ पे जो भी पीएच रहता है डिजोमीडिया विच अफेक्ट्स ऑन दैट डिजोल्यूशन। बिकॉज अगर आप पीएच एज पर एसटीपी नहीं किया तो वो डिजोल्व नहीं होते, तो उसका रिजल्ट कम आने के चांसेस रहते। So during preparation of dissolution media, you have to take care. Also, you have to degas your media, because if there are air bubbles found in media, then it affects the dissolution, uh, drug release. Then you have to there will be so much chances for contamination in dissolution, because dissolution टेक्निक में आप बफर यूज करते हैं। बफर यूज किया तो वो कंप्लीटली वॉशिंग के जब भी क्लीनिंग कर रहे हैं तो क्लीन नहीं होते, तो उस टाइम में वहाँ पे बिफोर स्टार्टिंग द एनालिसिस यू हैव टू गिव अ क्लीनिंग साइकिल विद मिथेनॉल followed by water, so that you there will कम चांसेस रहते हैं कि आप कंटेमिनेशन हो। तो बाद में वहाँ पे जो भी आपको मीडिया मेंशन किया है एसटीपी में 900 ml 1000 ml, you have to transfer that media in bowl and use paddle or basket. Basket is used only for when tablet is floated or for capsules. Also there is a sinkers are used for tablets. Some tablets are used sinker because their tablets are floated and not dissolved then. इस केस में आप सिंकर्स भी यूज करते हैं, यूज कर सकते हैं। लैडर और बास्केट ये नॉर्मली यूज करते हैं दोनों के लिए। बाद में वहाँ पे सैंपल कलेक्ट होने के बाद वो भी सैंपल फिल्टर करना है। डिसोल्यूशन का कम 100% फिल्टर करना है, क्योंकि अगर आप सैंपल फिल्टर नहीं करोगे तो वो वही इंग्रेडिएंट है जो भी है वो आपके कॉलम में चला जाएगा, और कॉलम है वो आपको जो भी सिग्नल दिखाएगा, जो भी पिक दिखाए उसमें इम्पैक्ट हो सकता है। तो वहाँ पे भी केयर लेना है कि आप जो भी सैंपल प्रिपेयर कर रहे हैं फाइनल वो फिल्टर करना है। तो यहाँ पे केयर लेना है डिसोल्यूशन में। और सेम एचपीएलसी में भी वहाँ पे यहाँ पे एचपीएलसी में जो भी हम सैंपल बनाते हैं वो भी विदाउट फिल्टर इंजेक्ट नहीं करेंगे, क्योंकि अगर फिल्टर नहीं किया तो आपको कॉलम में चला जाएगा, कॉलम खराब हो सकता है। और कॉलम इज द हार्ट ऑफ द एचपीएलसी, सो यू हैव टू टेक सो मच केयर ड्यूरिंग एनालिसिस। USP dissolution apparatus is one is basket, and in dissolution apparatus 2 is paddle. As per IP, 1 is paddle and 2 is basket, and temperature is same 37 degree plus or minus 0.degree. In disintegration it is plus or minus 2 degree. In dissolution it is plus or minus 0.5 degree because of our body temperature. Okay, थैंक यू। अगर आपका कुछ सवाल है तो आप पूछ सकते हैं।
Yes, sir. Thank you so much, sir, for your this nice lecture. You have nicely delivered your session, and this is a very interesting topic related with the instrumentation. Thank you so much, sir. You have just gone through the instruments which are used in the quality control department, like that HPLC, disintegration, dissolution, principles of all these instruments, as well as the different parts and the particular different parts of all these instruments. And definitely that will be really helpful to, especially while learning the quality assurance. One of the analysis, one of the pharmaceutical analysis, one of the subject is there in the third year. So definitely that will be helpful to understand the again basic things related with all this instrumentation. And based upon your session and your lecture, yes, definitely students, I hope they got the some particular information and the knowledge regarding this all these instruments and their role in the pharmaceutical industry. So sir, thank you so much, sir, honorable Mr. Srikant Gad sir, for your this nice session. So I would like to request or instruct to our students, anybody have any queries regarding these instruments which are used in pharmaceutical industry, and these are the analytical instruments which are used for the quality control in the quality control department. So yes, anybody may have any queries, so you can ask. Please feel free. Yes, anybody have any queries? Even you can write in the chat box, so you can put your queries in the chat box, so that will be again more interactive, and that will be more helpful to answer your queries. Yes, anybody how? Sate sir,
Yes, sir.
any students, any queries?
Any students from diploma pharmacy having any queries, please feel free to ask. Veer Dhaval sir is there, Gad sir is there, so you can ask anybody if you have any doubt in your mind
Yes, in this
regarding this
regard,
lecture.
yes. Sir, principal sir is there.
Yes,
Yes
Gautam
sir,
sir.
yes sir.
Yes, Jagtap sir is there.
Yes, any students, any have any queries regarding this? I hope yes, most of the students, they have cleared their doubts. Miss, we can say what they have cleared their concepts regarding all these instruments which are used in quality control department. So thank you so much, sir, Honorable Mr. Srikant Gar sir, and Honorable Mr. Veer Dhaval Kamble sir, for your nice
Durgesh sir, I would like
session.
to share.
Yes, sir.
Okay.
Please, sir.
Yes,
Yes,
sir.
sir. Over to you,
Please.
sir.
Yes,
Please,
yes.
first.
कांबळे सर आणि गाड सर तुम्हाला भेटायला खूप फिजिकली आनंद होईल. So I'd like to wish you to visit our Yashwanthav Hostel College of Pharmacy as well as D Pharmacy, both the unit, and suggest if you wanted to suggest something new or innovative in the our laboratories. Okay, we have machine rooms, instrumentation rooms. We have purchased the small unit for the tablet punching mini press also. We have DISO, electrolab, and disintegration and all these units. We are going to purchase very soon HPLC and IR very soon. We have right now UP. But till we have started this year M-Pharm Pharmaceutics also. So definitely साठी सर मी तुम्हाला पण सजेस्ट करेल की दोघी सरांना एक वेळ व्हिजिट करता आली पुढच्या महिन्यात किंवा जेव्हा ऑफलाईन कॉलेज सुरू होईल तेव्हा आपण करूया.
Yes, sir. Definitely. We will arrange.
डेफिनेटली जसं आपल्याला संजीव गोपी सरांचं पण बऱ्याच वेळा कॉपरेशन होतं आणि मुलांना गायडन्स होतं, तर आयड लाईक टू रिक्वेस्ट बोथ ऑफ यू गाड सर अँड कांबळे सर टू ऑलवेज शेअर युअर नॉलेज, करंट नॉलेज विथ आवर स्टुडंट्स. आणि डेफिनेटली माझ्या मते व्हर्च्युअल एक माध्यम आहे आणि मुलं पण आमचे एन्थुजियास्टिक आहे. सो सिन्स फ्रॉम द स्टार्टिंग ते तिथे होते. खूप छान प्रकारे तुम्ही दोघींनी माहिती सांगितली. गाड सरांनी पर्टिक्युलरली सर्व ॲपरेटस अँड डिसइंटिग्रेशन त्याचे डिटेल्स त्यांनी दिले. इवन आय थिंक HPLC आणि त्याच्यावर पण त्यांनी फोकस दिला. मोस्ट ऑफ ऑल डिप्लोमा फार्मसीचे आता स्टुडंट्स आहे, पण डेफिनेटली तुमच्या लेक्चरमुळे ते इन्स्पायर होतील आणि डिग्री करतील अशी आशा करतो. कांबळे सरांनी पण खूप डिटेल्स मध्ये आपल्याला रिलेटेड स्ट्रक्चरल फूड क्वालिटी ॲश्युरन्सच्या रिलेटेड माहिती दिली. आणि सकाळी पण मी सरांशी बोललो आता मगाशी, नानोस्कर सरांशी. त्यांचं पण खूप डीप बेसिक त्यांनी JMP ते इतक्या छान लेवलवर शिकवलं की एखादा मुलगा पुढे जाऊन एंटरप्रेन्योर पण होऊ शकतो, स्वतःचं एक स्मॉल स्केल युनिट ओपन करू शकतो. So really thank you to all you three. Some limitations are there due to virtual communications, but wholeheartedly our team will be more happy if you visit in coming next year. Okay, thank you to both of you, sir.
Yeah. Yes, sir. Thank you so much, sir. Now moving towards the conclusion part, at the end of this program, I would like to request Mr. Satyajit Sate sir, Principal, Yashwantrao Bhosle College of D Pharmacy, please deliver the vote of thanks. Yes, sir, please.
Thank you, sir, Gautam sir. Okay, on the behalf of Yashwantrao Bhosle College of Pharmacy and College of D Pharmacy, I am here to represent vote of thanks to all of you. First of all, I will thanks to Mr. Veer Kamble sir, Mr. Srikant Gad sir, who has worked in Glenmark Pharmaceuticals for last 15 years, and they have given us very precious time and valuable guidance to us. After that, I'm very much thankful to Mr. Sandeep Nanoskar sir for his valuable guidance and valuable presence. Okay, Nanoskar sir is physically present in college, visited our campus, and Jagtap sir, he is very happy to see our all labs and instrument rooms, and he is very positive to visit again to our campus. So thanks to Mr. Sandeep Nanoskar sir for their valuable guidance and precious time. After that, I am very much thankful to our honorable management, BKC management, honorable our executive chairman Achut Bhosle sir, who constantly support us and guide us. I'm very much thankful to our beloved principal, Honorable Dr. Vijay Jagtap sir. Uh, Due to his constant support and guidance, this event is made possible. And I'm very much thankful to Dr. Vijay Jagtap sir to provide us opportunity to deliver these programs or this type of programs. After that, I'm very much thankful to my colleagues, that is Omkar Pense sir and TPO coordinator, assistant professor Mr. Durgesh Gautam sir, who has all well coordinated this program. And because of to him, Omkar sir and Durgesh sir, we will make this event very successful and possible. After that, last but not least, I'm very much thankful to our students also, who has patiently listening, especially second year diploma pharmacy and final year B Farm and third year B Farm students, who has their patient listening and kind cooperation. So once upon, I'm very much thankful to all for making this event successful. Thank you, sir.
Yes, sir. Thank you so much, sir. Now, I would like to request all the participants, our honorable invited industry experts, and our principal, honorable Dr. V. A. Jagtap, sir, please share your video to click this photographs. Yes, please, sir.
Please on your videos.
तर शेअर अनस्क्रीन करा ना,
हाँ.
अनशेअर करा.
Guard, sir.
God, sir, just please,
हाँ.
you have to, yes, sir, please.
स्क्रीन शेअरिंग स्टॉप केला तर बरं होईल, सर. स्टॉप.
God, sir, hello.
Hello.
Yes, sir, please stop your screen sharing, sir, please.
सर स्क्रीन शेअर वरती क्लिक करा, तिथे तुम्हाला दिसेल स्टॉप स्क्रीन
हाँ,
शेअरिंग.
stop.
Yes.
Yes, sir. Thank you so much, sir.
Thank you, sir. Dear participants, please on your cameras.
Yes, dear students, all of you, just please share your, put your video on. Just a second. Yes, rest of the students, just please put your video on, all of you. Yes. 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 Thank you so much to all of you, honorable invited guest, industry experts, being present over here for this Rendezvous 2021. And I'm very much thankful, Honorable Dr. V. A. Jagtap, sir, our principal, Yashwantrao Bhosle College of Pharmacy. So your kind support to us. And at the end, I would like to thank all the dear participants, dear students of diploma as well as the degree. Thank you so much. Thank you, all of you.
Now I am declare here, so this program is concluded.
Yes.
Requesting all
the
leave
participants,
meeting.
yes, just I would like to share the feedback form. Just you have to submit your feedback form, so that will be a more helpful for improvement of future programs. Just a minute. Just I'm sharing the link in the chat box. So all are requested to submit your feedbacks, valuable feedbacks, so that will be more helpful for improvement in the future programs. So all are requested, you have to submit it. Just a second. So dear students, all the participants, please click onto the given feedback link and just you have to submit your valuable feedbacks. Thank you. Thank you all of you. After submission of your feedback form, you can leave. Okay.
devices.
Yes, all of you, once you will submit it, you can leave.